Our group is really interested in microbial products that get added to agricultural soils, and historically these have been really unpredictable in terms of whether they actually establish and function as we expect them to in agricultural systems. And so we wanted to ask, okay, when we add these products into a system and they're forced to interact with other organisms that are already established in the environment, what's going on, how are those other organisms impacting their function. And so that's what this system and the JGI project have allowed us to do, is look directly at how the presence or absence of underlying soil microorganisms impacts the activity of consortia that we add to soil surfaces. Ultimately, what we're hoping to do is determine sort of what it is that that makes inoculants or soil products effective in different agricultural systems. We'd like to be able to help develop products that are sort of more widely applicable, or improve products that are existing in certain ways by understanding more of the mechanisms that impact their success or failure as it relates to sort of interactions between organisms. Specifically, JGI is performing our metatranscriptomic sequencing, and so with those sequences, we're able to look at how the expression in the consortium changes in the presence or absence of underlying soil microbes. So we're able to look at specific genes that are being expressed under different scenarios. And so we can look at, for instance, are more genes that are involved in potentially in competition between microorganisms expressed when there's a more diverse microbiome present in the soil. So we're able to look very specifically at what's different in terms of how the consortium is functioning under different scenarios.